Good morning, everyone. My name is Andrew Nelson. I'm the Randall C. Pape Chair in Entrepreneurship and Innovation and Associate Vice President for Entrepreneurship and Innovation at University of Oregon. On behalf of the Phil and Penny Knight Campus for Accelerating Scientific Impact, it is my great pleasure to welcome you to this morning's Knight Campus Entrepreneurship event, and my great pleasure to welcome Professor Carolyn Bertozzi as this morning's speaker. Professor Bertozzi is the Ann T. and Robert M. Bass Professor of Chemistry and Professor of Chemical and Systems Biology and Radiology at Stanford University, and an investigator of the Howard Hughes Medical Institute. She completed her undergraduate degree in chemistry from Harvard University and her PhD in chemistry from UC Berkeley. After completing postdoctoral work at UC San Francisco in the field of cellular immunology, she joined the UC Berkeley faculty in 1996. In June 2015, she joined the faculty at Stanford University, coincident with the launch of Stanford's ChemH Institute. Professor Bertozzi's research interests span the disciplines of chemistry and biology, with an emphasis on studies of cell surface glycosylation pertinent to disease states. Her lab focuses on profiling changes in cell surface glycosylation associated with cancer, inflammation, and bacterial infection, and exploiting this information for development of diagnostic and therapeutic approaches, most recently in the area of immuno-oncology. Indeed, Professor Bertozzi has been a leader in not only conducting world-class research, but also exploring the applications of this research, including through several startup companies that she has founded or co-founded. Professor Bertozzi has been recognized with numerous honors and awards for her accomplishments. She's an elected member of the Institute of Medicine, the National Academy of Sciences, and the American Academy of Arts and Sciences. She's been awarded the Lemelson MIT Prize, the Heinrich Weiland Prize, and a MacArthur Foundation Genius Fellowship, among many others. Indeed, if I were to read a complete list of her accomplishments, awards, and honors, it would literally use all of our available time. So I'm going to stop here, but I will offer a quick note on logistics. After Professor Bertozzi's presentation, I will serve as moderator for the Q&A. If you have questions, please enter them in the chat function in Zoom, and I will then pose those to Professor Bertozzi. And with that, please join me in offering a warm virtual welcome to Professor Carolyn Bertozzi. Thank you so much for joining us.
Thanks, Andrew. I really appreciate that very kind introduction. It's fantastic to be here. I wish I was more here than on Zoom, but I'm happy that you have that background behind you. So at least I get to see the beautiful buildings and the campus that has sprung out from what was the last time I was physically in Oregon. It was just like a big open construction site. They had just cleared the ground, and I can't wait to go visit that campus in person at some point.
We'll be delighted to host you again anytime.
Great. So let me share the screen and tell you a story from my lab that has evolved over the last 10 years or so, which has to do with our interest in glycoscience. That's a longstanding interest of mine, and the intersection of glycoscience with cancer immunology and what we hope will be a new class of immune therapies. So the story begins with a tiny bit of background. I study glycobiology. That's the biology of complex carbohydrates. We also refer to them as glycans. And a lot of that biology is transacted on the cell surface. So if you just kind of take inventory of the plasma membrane and all of its constituents, there are proteins and lipids that project into the extracellular space, and many of them are scaffolds for glycans. They're glycosylated, and those glycans are biopolymers that encode information that allows the cell to interact with other cells in very specific ways. You can also see this cell surface glycans in my Zoom background. I call this my happy place. So I got interested in this field because of a long, basically half century or more of literature that associated changes in the patterns of cell surface glycans with certain disease states, and in particular cancer. So it's well known that the collection of glycans on the surface of cancer cells, they differ from the cells in the surrounding healthy tissue. And one of the most prominent changes that's been observed is an overproduction of families of glycans called Cyaloglycans. And they are called this because their terminal structure in the complex polymer, often branched glycan sequence is this monosaccharide sialic acid. So any glycan that terminates with sialic acid is a cyaloglycan. And it has been observed now time and time again that while normal healthy cells will have a certain pattern of cyaloglycans, I like to think of that pattern as like a well-manicured garden of cyaloglycans, but tumors that arise within the tissue often produce this sort of unkempt tropical rainforest of cyaloglycans. They're more dense in their distribution. Sometimes the structures are altered in certain ways. The things that the sialic acid is attached to might change. But there's this abundance of these cyaloglycans in tumor microenvironments. And that's been an observation. It's been a correlation. But until about 10 years ago, people in this field really didn't have a good idea as to why that might happen in the context of cancer. So things changed in the early 2000s when it became clear that our immune system plays an important role in protecting us from cancer. And when the immune system fails to do that, cancers can thrive. And that's a major underlying problem in people with disseminated disease. So your immune cells those are your white blood cells. They're circulating in your body, and their job is to patrol your cells that are in your body and determine whether they're good guys, healthy cells, or bad guys, diseased cells or foreign invaders. And the immune cells will sample the merchandise with a collection of receptors that are looking to engage ligands on that target cell, and information is collected through those receptor ligand interactions. And when a cancer cell is properly recognized as a damaged bad guy, the immune cells will kill that cell and get rid of it. And most of the time, this probably works just fine, which is why most of us spend most of our lives free from cancer. But of course, every once in a while, the immune system fails to do its job. And that's because the cancer cell has evolved mechanisms to escape recognition by the immune system. So zooming in cartoon form on what's happening at the synapse between the immune cell and the cancer cell. So immune cells have a collection of receptors that are activating receptors. Those are the receptors with a plus sign. Those receptors will recognize indicators of cell damage. And if they see enough of that, and those receptors cluster at this synapse, they'll signal to the immune cell that it's time to activate and do something to get rid of that cell. And it could be a direct kill on the cell, or it could be phagocytosis, like eating of that cell. It just depends on the type of immune system that's doing the job. But it's risky because if immune cells accidentally start killing healthy cells, that would lead to an autoimmune problem. So there's a checkpoint on the system, and those are what we call inhibitory receptors. So all of our immune cells have receptors that if they engage their own ligands at the synapse, they will deliver an inhibitory signal to the immune cell. And this inhibitory signal can quench the activating signal and prevent the immune cell from reacting. And that's important because it's a difficult job that the immune cell has to determine who's good and who's bad, especially if the bad guy is your own cell that's just been damaged, like a cancer cell. So, a cancer cell that happens to be able to express ligands for these inhibitory receptors is a cell that will survive the immune system. And so the phenotype of being able to turn off immune cells through contact, that's a phenotype that arises by natural selection during tumor evolution. So these inhibitory receptors have attracted a lot of attention as therapeutic targets. Probably the most recognizable family of inhibitory receptors are the so-called T-cell checkpoint receptors. One of those is PD-1. It's a protein on T cells that signals inhibition of the T cell. And another such receptor, CTLA4. These are very recognizable because they have been highly successful targets for immune therapies. In fact, targeting these two receptors is what put immune therapy on the map and why it became such an important practice in oncology. I thought I would just share with you a little bit more details of the T cell checkpoint biology. So PD-1 and CTLA4, those are receptors on the surface of the T cell. they are looking for signs of good health on the target cell, which would basically prevent the T cell from killing. So the cancer cell that can present ligands for PD-1, such as PD-L1 is what we call it, or ligands for CTLA-4, one such ligand being B7, the cancer cells that can express these ligands, recruit the checkpoint receptors, can deliver an inhibitory signal to the T cell. And that will undermine the ability of the activating T cell receptor, which is recognizing an MHC neoantigenic peptide complex on the cancer cell. It'll prevent the signaling. How it does that is through a battle here in the cytosolic face of the T cell between kinases and phosphatases. So the T cell receptor activates a kinase-driven signaling cascade, and these checkpoint receptors recruit through binding to domains of their cytosol that immunologists call the ITSM and the ITIM domain. These are just sequences of amino acids onto which phosphatases can dock. And the phosphatases at play here are called SHP1 and SHP2. These phosphatases take off the phosphates that the kinases had put on when the T cell receptor activated. And if you get enough phosphatase action at this synapse, you'll quench the signal and the T cell will just do nothing. It'll just leave and go to the next cell. So now, over the last decade, several companies have made antibody drugs that bind to either PD-1 or PD-L1 or CTLA-4 and block the ability of the inhibitory receptors to engage their ligands on the tumor cell. Blockbuster drugs like Merck's Keytruda, let me just shut my window, there's some noise outside, sorry about that. Keytruda is, Merck's #1 selling antibody drug right now. It's a PD-1 blocker. So is Bristol Myers Squibb's Opdivo, and BMS has an antibody called Yervoy, which is a CTLA-4 blocker. And then there's other companies with PD-1 blockers. Roche and Genentech have an anti-PD-L1 along with other companies. And the way this is unfolding is that pretty much every major pharma company has a PD-1 or PD-L1 or something like that blocking antibody that they are combining with their other therapies. So this checkpoint inhibition axis is becoming a really major component in the practice of oncology. And as you know, the importance of this biology was recognized with the Nobel Prize in Physiology or Medicine in 2018 to Jim Allison and to Suku Honjo. So why did people get so excited about this? It's because there were certain cancer indications that were basically, fatal diagnoses with no good treatment options. And among these particular indications, the observation in the clinical trials was that some fraction of them, a minority, but a significant minority, when treated with the checkpoint inhibitors, would not just have a survival benefit, they would basically have a cure. And to be able to cure even a minority of patients from a diagnosis that was otherwise totally fatal was really shocking and transformative. So here's an example of the Kaplan-Meier curves from this was the Yervoy clinical trial for a squamous cell non-small cell lung cancer. And you can see that the patients that were treated with the standard of care, eventually most of them passed away from, their disease. But in the patients that were treated with Yervoy, many of them also did not benefit from this drug. But at the tail end here, there was a survival of about 20% of the patients, and they just kept living and living and living and went into a durable remission. So this was amazing and transformative, but it also raised this really interesting question, which is, what was special about the 20 to 40% of patients who did respond to this therapy? And what was going on with the other 60 or 70%, the majority, who did not respond to this therapy? Was there some other mechanism of immune suppression or immune evasion that their cancers were eliciting that was not related to this checkpoint axis of biology? So this is where my lab, my students and postdocs, we would talk about these findings. And we had a hypothesis, which had to do, again, with this historic observation of overexpression of sialic acids on these tumors. Because back when the human genome was sequenced, it was found that we humans have a collection of sialic acid binding receptors that are expressed on all of our immune cells. And this family is called the sialic acid binding immunoglobulin-like lectins. They're Ig superfamily receptors. Cyalec is the abbreviation that hopefully will eventually become a household word. And in human, there are 14 of these receptors. So from the domain architecture standpoint, this is how we often represent them. They're transmembrane domain receptors, where the yellow line is the membrane. And outside the cell, they have various numbers of Ig domains, these little ovals. And they have this crescent domain is a sialic acid binding module that's called the VSET domain, for anyone interested. And you can see down here that each of these Cyalecs is expressed on different classes of immune cells. So B cells have Cyalec 2, also known as CD22. There's a chunk of Cyalecs in the middle here, Cyalec 7, 8, 9, 10, that are found on many different types of immune cells that are important for cancer immunity. So those include T cells, NK cells, macrophages, dendritic cells. And you'll note from this cartoon that many of the Cyalec family members have the ITIM domain and the ITSM domain. And just to remind you, those are the sequences of amino acids that are also found in the T cell checkpoint receptor, PD-1's cytosolic domain, that recruit the SHP phosphatases to the synapse. So these are receptors that have inhibitory signaling activity that's exactly the same way that PD-1 delivers its inhibitory signal. But these immune inhibitory receptors bind sialic acids. So we couldn't help but draw the analogy between what was known as an important axis of immune suppression for some minority of cancers, the PD-1, PD-L1 engagement on the T cell, and analogize that to the notion that maybe the Cyalec receptors, which do the same kind of signaling as PD-1, are inhibiting immune cell function on cancers that overexpress Cyaloglycans. And if that is true, and if that is an important mechanism of cancer immune suppression, it could explain this 60 years of observational data as to why cancers might evolve this phenotype. It would be a natural selection process to have an overabundance of sialicides. So we wanted to test this experimentally. And I won't go through the backstory because we've published on this now many times, but our very first entry into this field was to look at the effect of hypersialylation on the ability of NK cells to kill a cancer cell. So NK cells have a receptor called Fc gamma R3. That's a very activating receptor for the NK cell. And the ligand that receptor is looking for is actually an antibody that's bound to a neoantigen on a cancer cell. So, we have an immune response against our cancers, and that can include the generation of anti-cancer antibodies, and they can bind to cancer cells and allow NK cells to dock and kill. And that process is called antibody-dependent cell cytotoxicity. It's not only a natural part of our immune response to cancer, but it's also a mechanism by which antibody therapeutics can kill cancer cells. So the question we asked is, suppose that cancer cell has an overabundance of Cyaloglycans. In that setting, could a Cyalec receptor get recruited to the synapse with the cancer cell? And on the NK cell, that Cyalec would likely be Cyalec 7, which is one of those Cyalecs that signals just like PD-1. And if Cyalec 7 got recruited to the synapse, would that quench the activating signal through Fc gamma R3 and inhibit the NK cell so that it wouldn't kill the cancer cell? And sure enough, that's exactly what we observed. And not only was this a resistance mechanism against the sort of innate ability of NK cells to kill cancer cells, but hypersialylation also conferred resistance against some very important cancer drugs, like the antibody Herceptin, which binds HER2, which is a breast cancer antigen, and rituxin, which binds CD20, found on certain leukemias. Both of these are long-standing cancer medicines that work for some patients and not for others. We think the patients that don't respond are patients that have the hypersialylation phenotype to an extreme. So after that report, there were a number of reports that were kind of in related areas. There were other groups that looked at Cyalic-7 and Cyalic-9 in different cancer settings. There was reports on Cyalic-10 and Cyalic-15. And to make a long story short, there's now, I think, an overwhelming body of literature that would support the notion that the Cyalic-Cyaloglycan axis of immune suppression could actually be a dominant mechanism in human tumors, whereas the PD-1, PD-L1 axis might be actually a more minor contributor in the overall picture. If you want to read about this from a kind of high-level view, I thought CNE News did a really nice job writing a perspective on this field back in 2019. They named glycoscience a new breed of cancer immune therapy, so that was fun to see. So now, this being a lecture series dedicated to innovation and entrepreneurship, I thought I would talk about what do we do with this knowledge? And what we want to do is develop new therapeutics that target the interaction between the Cyalics and the Cyaloglycans for immune therapy. So how would you do that? An obvious idea would be to do exactly what Merck and company did to block PD-1 on the T cell, generate monoclonal antibodies that bind the Cyalics and prevent their engagement with their ligands. Now, in principle, that would be a simple pursuit, but in practice, it's complicated because there isn't just one Cyalic. This is a family of receptors. There are multiple Cyalics on multiple immune cell types, and it's entirely possible that you would need to block more than one in order to get, a deep effect on this axis of biology. So you would have to know which of the Cyalics do you need to block, and do you need to block more than one at a time? And many immune cells express more than one at a time. So it's complicated, and one would have to think that through. And quite honestly, I don't think the book on Cyalic contributions to individual tumors has been written yet. And so it's hard to make decisions at this point. The biology is still pretty early. On the other hand, you could say, well, what about targeting the ligands for the Cyalics? Could you disrupt them in some way? We thought that would be maybe a more interesting starting point, because even though we still have a lot to learn about the actual glycoproteins and glycolipids on cancers that support Cyalic binding, we do know one thing, which is that all of the Cyalics require, at minimum, a sialic acid residue as part of their binding determinant. And you can see this by looking at structures of Cyalics that have been solved in complex with different types of Cyaloglycans. And without picking apart all the details of what different Cyalics like to bind to, I'll just say that all of them have a hot spot in their glycan binding region, which is in the circle here, that's quite conserved across the family. And that hot spot engages the sialic acid. So, my students and postdocs, we talked through this and we thought, wouldn't it be cool if we could make a therapeutic that just strips all the sialic acids off of a cancer cell? Because that would deprive all Cyalics of their ligands in a Cyalic agnostic way. So from there was born this idea of what we called a molecular lawnmower, a therapeutic that we would target to the cancer cell surface and would literally just mow the lawn and take off the sialic acids and deprive that cancer cell of its ability to engage any and all Cyalics. So what molecule can act like a lawnmower? Our vision was to use enzymes to cut the sialic acids off. And there are enzymes called sialidases that catalyze that glycosidic bond hydrolysis reaction, and then fuse those enzymes to a tumor targeting antibody. And as a prototype, we started with Genentech's drug trastuzumab, which is an antibody that binds to HER2. And the brand name for that therapeutic is Herceptin. So this is an approved human therapeutic. My student, Melissa Gray, and a former postdoc, Hans Schau, who's now on the faculty at Rice, basically worked together to figure out how to and where to attach sialidases to this antibody. And one of their first prototype molecules was a fusion between an enzyme that we borrowed from Salmonella sialidase, that we conjugated down near the C terminus of the heavy chain here in the constant region of the antibody, keeping it as far away as possible from the parts of the antibody that interact with the antigen HER2. And their first generation molecule was actually made chemically. So my lab has a long-standing interest in what we call bioorthogonal chemistries. These are chemistries that one can use to do chemical reactions on proteins and biomolecules, and even in living cells, if the situation calls for it. So we used some of these chemistries and technologies to build this chemical fusion between the antibody and the enzyme. And these are also published chemistries, so I won't go through the gory details, but we used a site-specific conjugation method that we had developed years earlier called the aldehyde tag. And that allows us to genetically encode an aldehyde functionalized amino acid side chain into a protein at will. And so we encoded those aldehydes down near the C-terminus of Herceptin, and then conjugated at that site a bifunctional spacer, which had an indole on one side, and it's a functionalized indole that can do a Pictet-Spengler-like reaction with this aldehyde to form a carbon-carbon bond at that site. And on the other end, there's an azide. And of course, the azide is poised for what we call a copper-free click chemistry reaction with another molecule that has a cyclooctine. So to get the cyclooctine onto the sialidase, we just did some old school cysteine alkylation chemistry, and then just clicked together these two functional groups to make the final chemical conjugate. Now, the loaded question early on is, does this reagent selectively take the sialic acids off of the target cells? Those would be the HER2 expressing cancer cells. And it would not be good if this reagent was indiscriminately taking sialic acids off of all other cells, because we need those sialic acids there to help train the immune system of who's good and who's bad, right? So there's a reason most of our cells do have some sialic acids in a well-manicured garden. Those are signals to the immune system that I'm okay. So to test the selectivity, Melissa did this cool experiment where she mixed together in the same tube HER2 positive cells and HER2 negative cells, both of which had about the same level of sialic acid. And you can see how much sialic acid they have on this two-color flow cytometry plot. Here's the HER2 axis, positives and negatives. And this axis is the labeling with a lectin called SNA that binds sialic acid. So the y-axis measures how much sialic acid there is. And we took this population, treated them with the reagent, and then asked how much sialic acid is left on the two different cell types. And you can see these are different doses of the conjugate. Even at this lowest dose, sub-nanomolar dose, the cells that had HER2 expression, which were up in this quadrant, they fell down. So they've lost their sialic acids. And at about 1.2 nanomolar conjugate, they're completely stripped of their sialic acids. Whereas the cells lacking HER2 are completely untouched by this reagent until you get up into the triple digit nanomolar, where you start to see a little bit of off-target sialic acid loss. But it's a very selective reagent. Then the next question was, does the stripping of the sialic acids potentiate the ability of NK cells to kill these tumor cells? And the answer is yes. So what you're looking at here is how much killing occurred on a breast cancer cell line that expresses HER2 when those cancer cells have been mixed with fresh NK cells and one of the antibody reagents in our test set. And you can see that when trastuzumab has a sialidase on it, we call it T-Sia, you get very potent killing of these cells, more potent than what can be achieved just with the naked antibody. And remember, when there's just a naked antibody, the NK cell sees that antibody on the target and it wants to kill, but the sialicides will engage Siglec-7 and shut it down, right? So you get some killing with trastuzumab, but it's not as potent as when you have the sialidase. And in a really nice control, Melissa made another sialidase trastuzumab conjugate, but with a point mutation in the active site to kill the catalytic activity. And this sialic acid dead control conjugate is just like a naked antibody. And these are just some other controls as well. So that observation then motivated us to take these reagents into a mouse model of breast cancer. And for this experiment, we joined forces with my good friend Heinz Laubli, who's an oncologist at University Hospital in Basel, and his grad student, Michael Stanzak, had developed a syngeneic orthotopic breast cancer model in the mouse. It's the so-called EMT6 model, where one can inject these mouse breast cancer cells into the mammary fat pad and basically grow a tumor in a natural environment. And these cells have been engineered to express human HER2. So one can test HER2 type reagents in this model. It's also a model that is notoriously resistant to trastuzumab alone. And we recapitulated that observation here, showing that the tumors will grow. So this is days after inoculation, the tumors are growing and growing and growing. And whether those mice are treated with just a saline buffer or the naked antibody, those tumors just kind of take off. But when we injected those animals with the trastuzumab sialidase conjugate, now the tumors were very slow to engraft. And that's because they had been infiltrated with immune cells that are basically killing them along the way. So from this observation, we felt that it was time to try to translate the trastuzumab sialidase or antibody sialidase conjugate concept into a human therapeutic. So that's where the entrepreneurship element comes in. So I formed a company that is physically located in the Boston area. And at that company, which is called Pallion Pharmaceuticals, we have now developed a more drug-like conjugate. Instead of a chemical fusion of a bacterial enzyme with a human antibody, we now make genetic fusions with a human sialidase called human NU2. And we've got this construct organized so that that enzyme actually replaces one of the FAB arms, and the molecule overall has a molecular size that's very similar to a natural antibody. And we're all kind of on pins and needles to see how these molecules behave in humans. Right now, we're on track to get our first human dose of our lead compound in the first quarter of 2022. So the translation is underway. But meanwhile, we were frustrated because we felt we didn't have a very good understanding of the actual molecules on the cancer cell that is binding to different Cyalics. And there was some evidence from glycan array screens that although every Cyalic requires sialic acid as part of its binding determinant, the footprint it attaches to includes more than that. There's other sugars underneath the sialic acid, different glycan structures, and that maybe even some of the scaffolds beneath the glycan, proteins or lipids, could be part of those binding determinants. So we wanted to learn more about that. And toward that end, a postdoc, Simon Wisnowski, decided to use CRISPR screening strategies to get a deeper view of what are all the components of a ligand for a Cyalic. And he started with a deep dive on Cyalic7 and Cyalic9 because there had been so many associations with tumor immune suppression. So in this experiment, he took a cell line, the K562 cell, that had been transduced with a CRISPR single guide RNA library against every open reading frame in the human genome. And then we took soluble Cyalic FC reagents that were modified with fluorescent probes and used those as binding probes so that we could do a flow cytometry sort and sort away the variants that had lost their Cyalic binding from those variants that retained their Cyalic binding. And then the idea was, by deep sequencing, we would find all the guides that corresponded to a loss of Cyalic binding, and that would tell us which genes were important for Cyalic ligands. So here's a plot showing basically the genes that hit in the CRISPR screen for Cyalic7 versus Cyalic9. So any dot that is high on the y-axis is important for Cyalic9 ligand production. Any dot that is high on the x-axis is important for Cyalic7 ligand production. And all the dots here on the diagonal are important for both Cyalic ligands. And you can see that genes involved in sialic acid biosynthesis and Golgi trafficking to the plasma membrane, these are popping up for both Cyalics, as you would expect. But there were some really strong hits that were quite specific for Cyalic7 and not at all important for Cyalic9. And two of these, the pink dots here, are biosynthetic enzymes for O-linked glycans. And then this very strong hit, SPN, that's a gene that encodes a giant mucin glycoprotein called CD43. This was interesting because CD43 sort of has a sordid past. It's been associated with poor prognosis in cancer. It's a membrane-associated mucin, and by mucin, I mean it's a very heavily glycosylated, very tall, rigid glycoprotein where all these glycans that are shown as these diamonds, they're all on serine and threonine residues that are in these long stretches of clustered serines and threonines. And if you look through the clinical literature, CD43 was already well known as a poor prognostic factor in human disease. Here's one example showing the overall survival and event-free survival of patients with diffuse large B-cell lymphoma, and you can stratify their outcomes based on the expression level of CD43, where patients that have a lot of CD43 on their leukemias, they don't do well, okay? So we did a deep dive on CD43 because we had discovered it is a high affinity, prominent ligand for Cyalic. And what we discovered is that there's a very specific glycoform of CD43 that is engaging Cyalic. And all of the binding to Cyalic is concentrated in this one region near the N-terminus. Even though there's a lot of glycosylation, the only glycosylation that matters is way up here at the N-terminus. And it's this one particular cluster of serine and threonine residues that each of which carries a glycan called the dicyallyl T structure. It's a cool little O-linked glycan with not one, but two sialic acids on the same core disaccharide. We figured all this out biochemically, but I'm going to come back to the structure in a bit. Okay, so keep this in mind. Because in parallel, I had an MD PhD student who was interested in other questions. For example, we know that cancers often overexpress these cyaloglycans, but he was interested in what happens inside the cell that leads to this overexpression. Okay, so how does it happen that these cells become hyper-sialylated? And to understand this, we basically took inspiration from a publication that had come out of my Stanford colleague, Dean Felcher. So Dean is an oncologist who studies the transcription factor MYC. MYC, of course, is famously overexpressed in like over 50% of human cancers, well known to drive proliferation of cells. But it turns out that in addition to driving a proliferative program, what Dean had reported back in 2016 is that MYC can also drive the expression of immune suppressive proteins. And he published in Science that both CD47, which is a ligand for another immune checkpoint receptor, and PD-L1, the famous ligand for the T-cell checkpoint receptor PD-1, both of these proteins or their genes, the transcription of their genes is driven by MYC, at least in certain leukemia that he studies. So when we saw this paper, we couldn't help but wonder, could it be that an oncogene like MYC also drives the expression of cyaloglycan ligands for Cyalics, because that would be the parallel situation for us. So we teamed up with Dean. Here's Dean and his postdoc, Anya Deutzman, because they had all of these really nice transgenic systems where one could turn off or on the expression of MYC and look at the consequences in a murine tumor model. And he has this nice model of a T-cell acute lymphoblastic leukemia, where there's a transgene such that in the absence of doxycycline, MYC is constitutively expressed at high levels, that's the MYC on state. But if you add doxycycline, it engages the TET repressor and you can repress the production of MYC, and that's the MYC off state. So you've got these isogenic cells, which can be either MYC on or off with a chemical switch, and then you can compare them in different settings. So Benji Smith, the MD PhD student from my lab, basically took cells and turned off MYC or on MYC, and then compared their transcriptomes by RNA-seq. He compared their glycomes by mass spec glycomics, and he compared their ability to bind Cyalic Fc reagents. So that's a measure of Cyalic ligand production. And here are some data. It's quite striking. So first and foremost, he found that some glycogenes are very strongly regulated by MYC, and one that really stood out was a sialyltransferase called ST6 GalNAc4, which was already known in the literature biochemically as a disialyl-T synthase. In other words, this sialyltransferase can convert this monosialylated trisaccharide called Cyalil-T to the disialyl-T structure, which was precisely the glycan that we found concentrated on that CD43 ligand for Siglec-7. Not only that, when we looked at the abundance of different types of O-glycans on cells in the MYC on or off state, he saw basically a relationship of disialyl-T to MYC. So when MYC is on, disialyl-T here, the ion intensity of that glycan in the mass spec that's the most prominent glycan on the cells in total. And when the MYC is off, its abundance drops by 50% or so, and its precursor goes up, okay? So it was clear that MYC can regulate a change in cyaloglycan expression, and that this disialyl-T structure could be one of the major affected structures. But most importantly, we found that MYC could regulate the ability of Siglecs to bind to these cells. So what you're looking at here is a heat map, which basically represents the binding activity of the Siglec FCs in a flow cytometry experiment. So just looking at the top here, these are all the different Siglecs that we were able to get our hands on. They're human Siglecs, mouse Siglecs. Don't worry about the details. All that matters is that there's a bunch here up top, human Siglec-9 to some extent, very strongly human Siglec-7, some of the mouse Siglecs. Their binding to the TALL cells is basically controlled by MYC. So when MYC is on, their binding is high. When MYC is off, there's a significant reduction in binding. And what Benji did then is he knocked out that enzyme, ST6-GalNAc-4, that disialyl-T synthase. And what he found is there's a total loss of Siglec-7 binding on these cells in that knockout, and also a reduction in Siglec-9 binding. So this was a real revelation, because it suggests that this one enzyme, ST6-GalNAc-4, that's a switch that turns on or off the ability of Siglec-7 to bind to these cells through that disialyl-T structure. So we have this model now that MYC, when it's overexpressed, among all the other genes it regulates, which are involved in proliferation, it can activate the expression of ST6-GalNAc-4. And the consequence of that is there's an increase in the flux of this trisaccharide to a more highly sialylated tetrasaccharide. And when that builds up on the cell surface, an immune cell comes along to test whether this is a good guy or a bad guy. And if Siglec-7 engages this disialyl-T structure, it thinks this is a good guy, and that immune cell is inhibited. We tested this in a tumor model by simply taking those ST6-GalNAc-4 knockout cells and asking whether they could grow and thrive, in an animal compared to the wild type counterparts. And sure enough, the blue bars here, this is the tumor volume after several days after injection into the mouse. These tumors grow quite well when they have ST6-GalNAc-4 and when they express high levels of disialyl-T. But if you take out that enzyme and you take out that glycan structure, they have a much harder time in grafting. And that's because immune cells are in there. And we've looked at the immune profile of these cells, wild type versus knockouts. And yeah, there's definitely an effect on the immune system. So the implication from this is that ST6-GalNAc-4 could be a target for inhibition in the immune therapy context. And maybe one could develop a small molecule drug that inhibits this enzyme and prevents the biosynthesis of this immune suppressive glycan. And that's something that we're working on now. We're going to test that hypothesis. So in a nutshell, the takeaways from this lecture are: first, tumor cyaloglycans engage immune cell Siglec receptors and drive immune suppression. We had come to that conclusion back in 2010, 12 or so. And I think since then, there has been a lot of independent work that points in that same direction. So hopefully, the immune oncology world has now embraced this as an important target family for cancer immune therapy. And then more recently, we've taken a targeted cyaloglycan degradation strategy to interfere with this biology in a therapeutic context. And I hope that antibody sialidase conjugates, end up as a new breed of immune therapies at some point, and we're going to do our best to bring that to life. But then we've made some discoveries in the basic science area that I think also have translational implications. For example, I think the world is coming to the conclusion that all of these Cyalics are not redundant with each other. Each of our Cyalics has its own preferred biological ligands. And for Cyalic 7, we unearthed one of those through CRISPR screens, and it is CD43 in this di-Cyalic glycoform. So just knowing that could inform the development of new kinds of therapeutics. Maybe we could make an antibody against that particular kind of macromolecule and have a more targeted therapy that affects patients with a Cyalic 7 axis, for example. And then finally, I think there's a lot of interesting discoveries to be made in connecting oncogenes and oncogenesis with changes in the cell surface glycan pattern. You know, we found that MYC can drive the expression of a particular glycan, which is functionally important for immune suppression. That's interesting, but what about other oncogenes, right? KRAS mutants are prevalent human oncogenes. EGFR mutations drive oncogenesis. Do these oncogenes also affect glycan synthesis in a way that's functionally relevant for cancer, right? Those are open questions. We would like to answer them. And with that, I will thank all the people. I mentioned already the main players for each of these parts of the talk, but they're surrounded by a supporting cast that's also very important. Students, postdocs, undergrad staff, collaborators are always very important in the work that we do. So Heinz Laubley with his beautiful EMT6 HER2 positive mouse model, that was quite important. And then Dean Felsher and Anya Deutzman, who are teaching us all about MYC biology, which has, I think, a very rich intersection with glycobiology as well. And thank you once again, Andrew, and all of the folks at Oregon who invited me, and open the floor for questions.
Thank you so much. That was incredibly impressive and a lot of food for thought there. As I invite questions, please submit them through the chat window. But I'm going to kick things off. You went back and forth a bit between some of your quest for understanding, the big question of why, and why understanding some of the mechanisms, and then many of the applications which have come out of your work. I wonder if you could elaborate a little on that relationship. Sometimes we hear from faculty who really are driven by that question why, and sometimes a potential application appears and then they go down the path of starting the company. Other times we hear that really there's an eye towards application in the first place, which then informs subsequent basic research. And I wonder if you could elaborate a little more on your experience of how this relationship between fundamental understanding and application has played out in your work.
Yeah, that's a great question. You know, for me personally, I've always been, like many chemistry trained people, you know, I'm a chemist at heart. And as a chemist, I was always interested in what I could bring to biology in terms of tools and technologies. Like that was kind of I found myself gravitating towards those kinds of problems. In the earlier parts of my career, you know, when I first started my own independent lab, I was interested in developing chemical tools to basically solve some problems in basic research. You know, back then, you know, this was the mid 1990s, you know, I had been trained in carbohydrate synthesis as a PhD student. Then I had postdoced in an immunology lab. And it was an immunology lab studying some glycobiology. And in that lab, I was frustrated because the tools for studying glycans in living systems were so primitive at the time. There were so many things you couldn't do, and you couldn't ask questions, and everything was very laborious. And I remember thinking, this is awful. I mean, we need, you know, this is like an area where a chemist could really maybe bring some value, you know, and develop some tools. So where I started in my own career was to develop chemical tools for imaging of glycans, profiling, sequencing by mass spectrometry, right? Figuring out, like mining the glycoproteome, right? Just by figuring out the structures and what's expressed where. And so that's how I started. I wasn't really thinking so much about therapeutics or, you know, clinical translational science, but that sort of evolved for me as time went on. And it was, you know, back again, really, I would say this project was the project where, you know, we were sort of curious about, like, how is it that there's 50 years of observational literature about this sialic acid phenotype, but the the reason for this is not known, or the functional significance was not known, And so we started working on that. And simply because we had the insight that came from all the PD-1 biology was becoming hot, right? And we knew the Cyalics were signaling like PD-1. And so we're like, well, what if they're just immune checkpoints, right? Responding to Cyaloglycans. And once we got into the science there, then the translational work became obvious. It became obvious that there was a therapeutic opportunity there. And I wanted to pursue that. But I was at a place, I was at UC Berkeley at that time, and I was there for almost 20 years. And Berkeley is a wonderful institution, but it's a very basic science oriented place. There's no medical center at Berkeley, right? So chemistry is really strong, engineering is really strong. But if you want to do more translational science, you have to find your partners outside of the university. And I imagine at University of Oregon, you've probably had conversations like this, right? Because even though U of O has a medical center, it's in a different city, right? So where you are in Eugene, it's a bit isolated from all the clinical translational stuff. And I remember when the night campus was just a sparkle in the eyes of the people there, and you folks would talk with me about this early on. I remember there was the hope that you could bring some more translational science closer to Eugene through the appointments that you made in the Knight Institute. So I'm really curious to hear how that's gone. But for me, I had that same frustration that you had. And the opportunity came for me to move to Stanford. Stanford is an integrated campus with a medical center, hospital, physician scientists, clinical trials, patients, and chemistry, and biology, and engineering, basic science. So I pursued my translational aspirations by moving to Stanford.
Yeah, no, that's very interesting. We can talk more about that. Certainly, our partnership with OHSU in Portland is key to what we're trying to accomplish. I'm going to direct we have several questions now in the Q&A. Are you able to see these also, Professor Bertozzi?
Let's see, I do.
Okay, great. So some of these, my disclaimer here is my PhD is in engineering. So for Professor Karen Gilliman's question, which maybe we'll start with, perhaps if you could actually read that question and respond to it directly so that I don't ask it inappropriately.
Karen says, do you know the normal function of ST6-GalNAc4? Would an inhibitor to this enzyme have negative consequences? This is a great question. So what's the normal function or the disiallyl structure, right? Why do we make it? Not much is known. So there's like 20 sialyltransferases in the human genome. This is one of them. Some of them have been knocked out and phenotypes have been characterized, but this is not among them. So there is no global mouse knockout that's been reported for this enzyme, which would sometimes that can be revealing, of a natural function. It is not, if you just look at like old northern blot experiments that have been done before RNA-seq, people just did northern blots on tissue lysates. It's not that widely expressed. It might be an enzyme that is more developmentally regulated, which is true for some other sialyltransferases. That meaning some of these things are expressed at high levels during embryogenesis, but not so much in the adult. And I think it falls into that category, which means it may have functions it actually could be driving an immune suppressive program during embryogenesis. Cancers and embryos are sort of similar in many ways. There's a lot of proliferation, and whenever there's a situation of a lot of proliferation, there's a risk of immunogenicity, which is why you often see immune suppressive programs accompanying things like wound healing, right? Or, embryogenesis. So I don't think I have a good answer to that question. This is not an enzyme that has been widely studied.
Future opportunities.
Yeah, right.
Let's turn to Bob Goldberg's question. Bob is the director of our Knight campus for Accelerating Scientific Impact, and he asks, what type of cancer do you think your targeted degradation of cyaloglycans will be best suited to treat? Yeah. Does this therapy have the potential to treat solid tumors?
That certainly would be the hope. So right now at Pallion, they've developed a suite of Cyalic, sort of soluble Cyalic reagents that they use to do histopathology on tissue sections, on biopsied material. So just like we use those Cyalic FC, those soluble probes to sort of probe for the presence of ligands for the Cyalics, they made a kind of souped up version of that. That's a very, very tight binder, and they have shown beautiful histopathology data. So you can take tumor sections from the biopsy, stain them with these reagents and see, do they express high levels of Cyaloglycans? This is exactly how people score tumors for PD-L1 expression or HER2 expression or any of these molecular markers that would then lead to, the prescription of the drug. So the idea is that we would analyze the tissue, use the Cyalic reagents to see if they're like high expressors of Cyaloglycans. And if they are, you would stratify those patients for treatment with these therapies. And I can tell you from some of their data, I think they actually presented these data at the last AACR meeting. So they're public data now. But melanoma patients, lung cancer patients, there's certain leukemias, ovarian cancer patients, colon cancer patients. Those are solid tumor indications, except for the leukemias, where you get a prominent, more than 50% of the patients are screaming hot with binding to these Cyalic reagents. So, but that right now is the strategy, is to look at the tumor tissue, see if they have a lot of Cyaloglycans. If they do, put them in a clinical trial. Hopefully they're going to be the responders.
Thank you. I'm going to jump to Paul Dalton's question next. Paul was one of our Net Campus faculty members, and he asks, is there a need to establish 3D cell culture to advance this research? I assume that the in vitro work was performed in 2D. Are there implications within vascularized 3D systems?
That's a great question. Yeah, I would echo what I think is the underlying sentiment, that if you're studying glycobiology or any sort of cell surface interaction phenomenon, to do it in 2D is to do it at risk. It's at your own, you might perish for that pursuit. But so for our work, we, in our in vitro work with these HER2 reagents, all of that was done on tissue culture plastic until we went into the mouse. I think tissue culture plastic and mice, neither of those are very good models for human disease. Three-dimensional cell culture or organoid cultures, these are better models for human disease, but they bring with them obviously some, their impracticalities. But I think it would be great to have good organoid or the minimally 3D cell culture models for some of these experiments. And I think particularly for the MYC experiments, because, how oncogenes regulate transcription is very sensitive to the tumor microenvironment. Right. So, I would love to try these in vascularized 3D systems that emulate the the immunological part of the tumor microenvironment. That would be very cool.
Awesome.
Do you have such a system? We'd be happy to send some reagents.
It sounds like a subsequent conversation with Paul, perhaps.
Yeah.
Let me combine the next two questions, which were started by Chris Liu, who's one of our business school faculty members, though his first PhD is in biology. So he asks, you mentioned Pallion is headquartered in Boston rather than the Bay Area, which I found intriguing. What are the costs and possible benefits to doing a long flight? And a subsequent question, I'm interested in knowing how you developed your collaboration with a Boston-based company. Are there mechanisms at Stanford that help that sort of relationship?
Yeah, I think I'm on my 8th company now. Pallion was somewhere in the middle of that string. And every single one of those companies had a different backstory formed in a different way. So the Pallion backstory is that I had just moved to Stanford, and it was really only 2 months after I moved. We were still setting up the lab, and we had published that paper on the sialic acids engaging the Cyalics and a hypothesis that this was a big, new target class for immune therapy. And the CEO of Pallion, whose name is Jim Broderick, he at that time was an entrepreneur in residence with SR1, which is the GlaxoSmithKline venture arm. And they are headquartered in Cambridge, Massachusetts, and he was a Boston person. So that's where he was located. And they had given him the mission, and this takes me back now to about 2013, 2014. That's when he joined up with SR1. And he was a serial entrepreneur, had been a successful CEO with a few other biotech companies. And they basically gave him $2 million of seed money. And they said, go find for us the next big thing in immuno-oncology, right? This was right after Keytruda had been approved, and it was like all the rage, and everyone wanted to get in on it and find the next thing and get the other 80% of patients to respond and so on. So he spent a year just like looking around at stuff. I mean, he just was talking to people, KOLs, all over the world, and thinking about, he himself had background in immunology. He's an MD, So he was a physician trained. And somehow he came to glycoscience. So, and then somebody said, oh, you should talk to Carolyn, right? So he cold called me and he said, here's what I'm trying to do. I'm looking for something. And someone's, I hear that you're sort of interested in this, and can we talk? He didn't know at that time that we had been making these sialidase conjugates, because we had just started that at Stanford. And I showed him what we were doing, and I said, this is what we think. And after a few days, he called me back and he says, I want to start a company around this. Will you join me? Right. So he sort of provided the startup energy and the seed funds. And then we pulled in another co-founder named Paul Crocker. Paul is one of the immunologists who discovered the Cyalics in the first place. Paul is centered at University of Dundee in the UK, right? So you've got me in California, Paul in the UK, and Jim in Boston. And since Jim was going to be the full-time CEO, his company, it really made a lot of sense to do it in Boston. And it wasn't totally unpalatable for me, because I grew up in Boston and my parents still live in Lexington, and this company is in Waltham, and it's about a 10-minute drive. So, I would visit my parents, I think we all learned during COVID that maybe it doesn't matter so much where your company is physically located, unless you are there at the bench, right? And I, as an advisor to the company, it's just fine for me to be on Zoom.
That's fascinating. And sadly, that brings us to our time. We didn't get to Sam Lester's question. He was one of our bioengineering PhD students, but you will see him later when you meet with our PhD students. So let me close by first, of course, thanking you so much. This just so inspirational. I think we could have gone on much longer, and we have many more questions. So I'm looking forward to our meetings later today. And I also want to thank all of our participants. This is our final event this year in the Entrepreneurship Speaker Series, but I do want to point you towards some of our other events coming up. Most immediately, this Thursday on April 29th at 1 p.m., we are very pleased to co-host Alondra Nelson. She'll be speaking on the social life of DNA, race, reparations, and reconciliation after the genome. And Dr. Nelson was recently appointed as Deputy Director for Science and Society at the White House Office of Science and Technology Policy. So for details on this and other upcoming events as part of the Night Campus, please visit our webpage, accelerate.uoregon.edu. And thank you again to Professor Carolyn Bertozzi. Just a fascinating and inspiring talk. We so much appreciate your time with us.
Thank you so much.